All right, hi everybody.
Hi, Dr. Barker.
I hope that you are all doing well. So just a couple of updates. My guess is I'm probably going to end up doing the exam grading this weekend. Maybe it'll happen before then, but my guess is this weekend, if I'm being 100% honest with you. So just that's the story with that. You'll know on Friday if that changes, but that's where I think I am with stuff. You got an email from me yesterday that I updated the course schedule. I originally had us having 2 primary lit assignments, one of them being due on Friday, and so I was actually working on this to put together that assignment. And then when I looked, I was like, well, wait, how many points is it worth? And so I looked at some gradebook stuff, and I realized that, in fact, in the gradebook, I only had 1 primary lit assignment, and on the syllabus, I had 2, which meant I got to cut one. So we don't have one due this week anymore. So that is off. I made one other little schedule tweak and put up an updated schedule on, I think, yesterday. So that is the plan there. Today I'm going to talk about peripheral B-cell responses, and then on Friday I'm going to talk about response kinetics and memory. And so these are all really good kind of key important topics in watching our immune response happen with some kind of microbe. So previously, I left you on a cliffhanger with B cells. So we talked about B cell development in the bone marrow. We saw processes of B cell central tolerance after our B cells did development. This is all happening in the bone marrow, this yellow location in the body here. And then I told you that if the B cell is successful in making a B cell receptor that is not self-reactive, the B cell leaves the bone marrow and goes into the periphery. And then I never told you anything else about what happened to that B cell. And so today we're actually going to pick up with that B cell and see details about B cell responses in the periphery. So we're in those same pink organs, our secondary lymphoid organs like the lymph node and spleen, potentially also others like the Peyer's patches, where our B cells are going to respond to the periphery. The B cells can get to those secondary lymphoid organs using the same kinds of trafficking processes that we saw for T cells. So I don't need to tell you anything about how the B cells are getting there. I also don't really need to tell you about how the T cells are getting there, because we already talked about that. So we've got those naive T cells coming from the thymus that might come into the HEV and then come into our lymph node or spleen, our secondary lymphoid organ. B cells are doing the same thing. Just the T cells are going to move to the T cell zone, listed here as T cell area, and B cells are going to go into this B cell zone. We also, if you remember, have our dendritic cells coming to this same location, potentially after picking up antigen. So we have some dendritic cells that have come to this same location. They're hanging out in the T cell zone, ready to interact with the T cells. And we have one other thing that's going to be important for us today that will come into the secondary lymphoid organ or the lymph node, and that is free antigen. So we've seen some antigen coming into the secondary lymphoid organ with our dendritic cell. But there also might just be antigen proteins floating around in the lymph that get carried into the lymph node. And so remember that when we think about the lymph, random proteins or whatever is in that liquid outside of cells is getting pushed into the lymphatics. And so we're going to get just random antigen pieces also coming into this area. So the secondary lymphoid is going to be a key location where we're going to see all of these important players that we see today meeting up. We need to think a little bit, we've talked about the T cell, we talked about the dendritic cell, we're getting ready to talk about the B cell. The one other piece that we need to think about a little bit is the free antigen. So that free antigen that may come into the secondary lymphoid organ in terms of coming in through the lymph. We've already talked with the T cell about the fact that the T cell goes to the T cell zone. The dendritic cell goes to the T cell zone. The B cells are going to go to the B cell zone. So the thing that we have to figure out is, where does the free antigen go? And here in this slide, you can see that free antigen. So you can see these little like black little shape things that are just random antigen proteins or whatever antigen particles are coming in from the lymph that may come into our secondary lymphoid organ. And that free antigen, you can see kind of floating around all sorts of different parts of the lymph node. Just going to kind of float around until eventually it is going to bind to a specific type of cell called a follicular dendritic cell, or an FDC. And so you can see the follicular dendritic cell here, and you can see that the antigen, that free antigen, is going to bind to the follicular dendritic cell. Follicular dendritic cells have one thing about them and their name that is really good, and one thing about them and their name that is really bad. So if you had to guess where the follicular dendritic cell lives, where might you guess the follicular dendritic cell lives? Yes? In a follicle. It's got the name follicular dendritic cell. And that is correct. Follicular dendritic cells are found in a part of the secondary lymphoid organ, which is called the follicle. We also often talk about the follicle as the B cell zone. So we've got the follicular dendritic cell in the follicle. Yay. But then there's also the bad thing about follicular dendritic cells. Follicular dendritic cells are not dendritic cells. They're not myeloid. They don't do MHC class 2 presentation. They don't do phagocytosis. They don't do any of the things we talked about with dendritic cells. They're basically like a connective tissue almost. And so yes, they're in the follicle. No, they are not dendritic cells. You can see an example of a follicular dendritic cell on the right here. And so if you look at that follicular dendritic cell image that you see on the right, what do you notice about the follicular dendritic cell? Yeah, Jazz. Has lots of spikes on it. Kind of looks like, a thing with dendrites, right? Just like the dendritic cell did. And so, in fact, these got named dendritic cells because of how they looked under the microscope before anyone did any experiments to find out that they weren't dendritic cells. So they got the name follicular dendritic cells, and then someone did some more experiments, and they're like, oh, no, that just looked like it. It isn't actually a dendritic cell. But the key thing that we see when we see that follicular dendritic cell is it does have all those spikes, just like the dendritic cell does, and they kind of have the same reasoning, same function. In this case, it's really about having a lot of surface area. So these cells have a ton of surface area. But this free antigen that we're talking about doesn't go inside of the cells. We never internalize that free antigen. The follicular dendritic cell, which is in the follicle, here's the FDC in blue, has all sorts of receptors on its surface. Here we're seeing a complement receptor, but there's actually way more than just complement receptors. Basically, when I think about a follicular dendritic cell, sometimes I think about fly paper, that little sticky paper that people like hang from the ceiling to make flies fly into it. And the whole point of that paper is just that it's sticky. And so if something goes by it, it gets stuck. Follicular dendritic cells have so many receptors all over their surface that they're basically really sticky, and any antigen that comes by will bind. So follicular dendritic cells have complement receptors. So if your antigen has complement on them, that'll make them get stuck. Follicular dendritic cells have all sorts of kind of receptors, and the idea is that basically they're not going to have any antigen like go inside. We're not presenting on class 2, so we don't usually talk about follicular dendritic cells presenting antigen. They're just like a structure that's getting stuff stuck all over it. And so we have these cells with this big surface area covered with receptors that are basically just getting this antigen, which you can see here as like this little red thing, stuck all over the surface. And so sometimes people talk about follicular dendritic cells being decorated with antigen because they're just like covered with all the antigen that got stuck as it was flowing through the lymph node. And those follicular dendritic cells and the antigen on the surface of the follicular dendritic cell will be really important in thinking about what's happening with our peripheral B cell. So the next slide is really why we are talking about all of this here in this part of the course. When a B cell gets activated in the periphery, it can be activated in a couple of different ways. One of those ways is a T cell independent activation. So there can be situations where the antigen just activates the B cell in T independent, it's listed here as TI, T independent activation. That is okay. It usually involves some other signals in addition to the B cell receptor, and the other signals could be lots of things. So you can see a couple different examples here. But this kind of activation of our B cell in the periphery is really only just okay. The B cell gets turned on, especially if there's a lot of antigen, but we're not going to see class switching in this case. So we're not going to get anything other than that original IgM or IgD. We're not going to get the IgG or the E or the A in T cell independent activation. We're not going to get this process of affinity maturation where the B cell actually starts to bind to antigen better and get stronger at binding to antigen. That doesn't happen if there's no T cell around. We're not going to actually have that B cell really be turned on to become a memory cell to remember this encounter. B cell is kind of turned on in a meh kind of way. They always laugh at me on Twiv when I say different things are meh. That they're like, that's how we know you're younger than us. So that can happen with the T-independent activation, and T-independent activation does exist. Though I am really going to say almost nothing else about it this semester. But yeah, that can happen. But most of the time, or at least for the responses that we are going to talk about, in order for our B cell to become fully activated, the B cell needs a signal from a T cell. So generally, if we want to get class switched antibodies, so if we want IgG, which is like our really good antibody, or IgA or IgE, we have to have a T cell involved in activating that B cell. Class switch only happens in T cell dependent B cell activation. If we want affinity maturation for the antibody to get stronger and it's binding to antigen, that only happens when we have T cell dependent activation. If we want to have memory for this antigen, that only happens when the B cell gets T cell dependent activation. So really, for the responses we're going to be talking about this semester, our B cell really needs a T cell in order to be activated. And that's why I couldn't tell you all the details of the B cell story until we had the details of the T cell story. Yep, Jazz. No. So we're going to see affinity maturation more in later in this lecture, but basically the idea is that the receptor's affinity for its ligand actually changes and gets better. It matures. Yep. You can actually also see this. I'm going to go to the next slide and then I'm going to come back to this one. You can see this process happening here. If you want to class switch, so here you can see IgM and here you can see we class switch to something else. If you want to class switch, you have to have T cells involved in your B cell activation. If you want affinity maturation, there's also this process called somatic hypermutation, which is very similar to affinity maturation. We'll get there. If you want either of those things, you need T cells. If you want your B cell to really proliferate well, you need T cells. If you want to make a memory cell, really, if you want any kind of good antibody response, good classic antibody response that you would think about when we think about B cells, you need a T cell involved too. And so we're going to be talking a lot about T cell dependent B cell responses, which you can see here. And what you can kind of already see in this slide, we will say more about this on some future slides, is a little bit about how this process is working. And so what you can notice is that signaling through the B cell receptor is basically a signal 1 for the B cell. See, it has a 1 next to it. The B cell also needs a signal 2 and a signal 3 to be fully activated, just like the T cell needed a signal 2 and potentially a signal 3. The difference is in this case with the B cell, the signal 2 is coming from the T cell. So the signal 2 is not coming from an antigen presenting cell over here. The signal 2 is coming from a T cell. It's actually coming from a T cell that is seeing an antigen on MHC class 2 from that B cell. So we'll get into this a little bit more. And the signal 3, the cytokine, is also coming from that T cell. So here we've still got signal 1, signal 2, and signal 3. It's just that signal 2 and signal 3 are coming from this other cell partner that wasn't involved in this process before. You can also see, and I'll say this again more later as well, that signal 2 in this case on the B cell is a protein called CD40, while on the T cell it's CD40 ligand. So the B cell needs a CD40 signal that from the T cell showing it CD40 ligand. That's basically just like the CD28 in the case of the T cell that was binding B7 on the other partner. So we can kind of go through the steps of what is going on in this process. So the first thing that is going to happen is that our B cell is going to encounter antigen in the lymph node. So remember, we've got that free antigen sort of floating around free, it's called free antigen for a reason, in the lymph node. We also have some of it binding to follicular dendritic cells, but it's still on the outside of follicular dendritic cells, so the B cells could interact with it still. So the idea is that B cells are going to somehow encounter antigen in that lymph node or other secondary lymphoid tissue. And you can see this encounter here where the B cell is using its B cell receptor to interact with antigen. We've already seen another situation where B cells encounter antigen with their B cell receptor and something happens. That example was when we talked about MHC class 2 presentation by B cells. So if you remember, B cells are one of our professional antigen presenting cells. B cell, but they're not able to do phagocytosis. They're not a phagocyte. So B cells have their own way of internalizing antigen, and it was this process known as receptor-mediated endocytosis. So basically, when something binds to the B cell receptor, the B cell pulls that receptor into the cell. It endocytoses the receptor and whatever that receptor has bound, whatever that receptor has caught. That thing, that antigen that has been bound by the B cell receptor will get degraded in the endosome while the B cell receptor gets recycled back up onto the surface. So basically, when a B cell encounters an antigen with the B cell receptor, we're going to endocytose that antigen. This is also giving that B cell a signal 1. And now that the antigen is endocytosed, that antigen can be presented on MHC class 2. So the B cell is going to bring in the antigen, degrade it, and present it on MHC class 2. This will also sort of start to activate the B cell, but the B cell won't be fully active yet. You can also kind of see this process here. You can see our naive B cell entering into the lymph node through the HEV and heading to the follicle, which is just another word for the B cell zone, and potentially encountering antigen on the FDC or encountering some free antigen. When that process starts to happen, one of the things that the B cell will do is that the B cell will start to present this antigen on MHC class 2. So that's one thing, is this MHC class 2 presentation. The B cell also moves in the lymph node, however. It goes to a different site in the lymph node. And specifically, the site where the B cell goes is right to this border between the B cell zone and the T cell zone. So our B cell is now going to hang out right on the border between the B cell zone and the T cell zone. You can also see that the B cell may undergo a little bit of proliferation, though the proliferation is not like crazy important right here. It will become important later. There will be proliferation later. That's going to be a real big deal. This proliferation, not as big a deal. So you can see, here's our B cell. It encountered antigen. It's presenting on class 2, which we can't see here. It's moving to this border between the B cell zone and the T cell zone. While all of that was happening, the T cell also made its way into the lymph node, interacted with a dendritic cell, and got activated. And so here you can see we've got all these activated T cells hanging out with this dendritic cell. They've proliferated. See, there's way more of them than there were before. We've got our B cells hanging out here at the borderline between the T cell zone and the B cell zone. What will then start to happen is those T cells, some of those T cells will actually move to this border as well and start interacting with the B cells. So we're actually going to see some T cells also go to this border site to interact with the B cell and actually provide help to the B cell. And it's that help that gives us T cell dependent activation and all the types of activation that we were thinking about previously. And so for our B cell to get fully activated, for it to class switch, do affinity maturation, and make a memory cell, that B cell is going to need to get T cell help from a T cell in the lymph node. So you can see here's that B cell. It's internalizing antigen. It's now going to present on class 2. It's going to get some T cell help from a T cell that has been activated in the lymph node. One thing that you should know about this T cell is that this T cell is probably is a CD4 because it's recognizing class 2. I also told you it was T cell help, and CD4s are helper T cells. So we've got our CD4 helper T cell, labeled as TH here, that's interacting with this B cell. This cell has been activated. It's previously interacted with a dendritic cell, so it's now active. It's ready to go. It's making cytokines. It also has CD40 ligand on its surface, CD40L, so that it can give the B cell the ligand it needs for CD40 to get signal 2, and potentially give that B cell some cytokines in order to give that B cell its signal 3 of cytokines. And once the B cell actually gets all of these things from the T cell, that is going to kind of change a little bit about what's going to happen with the B cell. And so we're going to have some more events that are going to happen to the B cell once it interacts with a T cell, and that's how we're going to get all those good bonus functions: the affinity maturation, the memory, the class switching. When I originally learned this, when I started graduate school, this is kind of how we talked about it. We have now learned some additional information about what's going on with this process. So here we have the B cells kind of moving to this region of the border between the B cell zone and the T cell zone. What was the other word for the B cell zone? The follicle. And there have to be some T cells that are going to move to interact with these B cells. You are going to imagine, and we used to just say it's some T cells. And then we learned that it was a specific type of T cell, a specific subset of T cells that do that. So what specific subset of T cells might move to interact with the B cell here? Yeah. What does TFH stand for again? T follicular helper cells. So if it was like a Th1 cell, it's going to just go do Th1 stuff. But if it's a T follicular helper cell, it's going to move towards the follicle and do some helping, just like its name, T follicular helper cell, says. And so the really important cell, so again, we used to just say it's a CD4 T cell, but now we know it's a T follicular helper cell. And so those T follicular helper cells, one of those subsets that we talked about before, can be really important in helping B cells. This should be another way that you see that the T cell must have been previously activated, because it's not a T follicular helper cell to start with. It only becomes a T follicular helper cell after it interacts with the dendritic cell, and the dendritic cell kind of sets up what type of T cell activation happens. And so what we will see is we will see our B cell will be doing its internalization, which you can see here. And you can then see we're going to present antigen to the T follicular helper cell. That T follicular helper cell is going to provide CD40L to bind to CD40 on the B cell. That's giving the B cell signal 2. And the T follicular helper cell is also going to produce cytokines. And this is how we're going to get that awesome T cell dependent B cell activation. And you can also kind of see this process happening here. So here are the B cells, they're moving to the border. Now here are the T cells also moving to the border, interacting with the B cells. So the T cells are going to move to the follicle. We're going to see this interaction between the B cell and the T cell in the secondary lymphoid organ. And now the B cell is going to move again to another spot in the secondary lymphoid organ. Sometimes students really want a definition of what this spot is. And the definition of the spot, which I'll define in the next slide, but is the place where the B cell goes. So like the spot where the B cell goes is the spot where the B cell goes, is kind of how the definition works. The name of this place is the germinal center. So now our B cell, after it's interacted with a T cell, is going to go to a germinal center. Did the germinal center exist before the B cell got there or not? Is a whole area of debate with so many papers that we are just so not going to deal with it. So you may have that philosophical question for yourself. But the idea is that once this B cell has interacted with a T cell, it's going to move to the germinal center. And so it's going to participate in a process that's called the germinal center reaction, because it happens in the germinal center. there's one thing I can tell you about the location of where that germinal center is. So the one thing you should know about a germinal center is that a germinal center is in a location where there also is a follicular dendritic cell. So there has to be an FDC there as well. So the B cell is moving to a germinal center. There's also an FDC at the germinal center. And so you can see this happening here. And oh my gosh, this is also where the antigen went. So you can see here's my activated B cell. It's moving to the germinal center. So first it comes in the HEV. It sort of starts to interact with T cells. Then it's like, oop, up here to this germinal center. So you can see it making this first interaction here, and then doing the second little arrow here, going to the germinal center. So it made two moves inside of the lymph node. You can also see a germinal center in this cartoon at the bottom, and all I want you to notice right now is that there is an FDC in there somewhere. This is probably somewhere in the follicle. You will also learn, as we see today, that the germinal center can be divided into two areas, one called the light zone, one called the dark zone. And you can actually not very well in this lighting see a germinal center happening in this mouse. And so you can't see the green B cells at all, but they're naive, so that's okay. You can see after there's some antigen, we've got sort of some B cells coming in. Suddenly those B cells are sort of going to sort out to this blue area, which has a stain for the B cell zone. So you can see, eventually the B cells are all going to the B cell zone. And then you can see they move again into additional locations. And they will eventually form this thing called a germinal center. The germinal center involves a follicular dendritic cell. I already told you about follicular dendritic cells. Hooray. And so now that we have our B cell in the germinal center, we are going to see the rest of the steps of what that B cell has to do in order to become fully activated. And I'm not a B cell person. I apologize in advance to my sister listening to this recording because she did her PhD on B cells and she always yells at me for being insulting to B cells because I am not a B cell person. But this process that happens in the germinal center is actually pretty cool. And so we're going to be seeing this process that happens in the germinal center, which is also known as the germinal center reaction. When our B cell, comes into the germinal center, so here you can see our naive B cell, it gets activated through T-dependent activation. It's going to move into the germinal center. It's going to first come into the germinal center in the dark zone. And so you can see that shown here as the dark zone. The first thing that happens when this B cell comes into the dark zone is that the B cell is going to proliferate, aka divide, and make many copies of itself. And you can kind of think of this as yet another pat on the head. It's like, you are useful, make a clone army. Make many copies of yourself because you are useful, is what's happening here. If you look under the microscope at the two different parts of a germinal center, I don't know if you can see them quite as well here as I would like. You can't see it I'm not loving this image as much on this. But as I said, there's a dark zone and a light zone. Why do you think one zone might be dark? in this organ if you look at it under a microscope? Yeah, Roxy? Because there's a lot of cells there. It's super crowded. And that is why the dark zone is dark, because it's super crowded with a lot of cells. Why do you think the dark zone is crowded with a lot of cells? Yeah, Emily. That's where proliferation's happening. That's where the clone army is getting made. So basically the cells are going to move into the dark zone and start proliferating and make it dark by making many copies of themselves, making it really, really, really crowded. While the cells are doing this proliferation, there is another process happening. I'm going to say that this process is happening while the cells proliferate. I'm also going to say that it's happening, that there's another process that's happening at the same time. I'm going to say the proliferation and two other things are happening at the same time. This is another area of hot debate of exactly the right order between them and whether they're exactly at the same time or what. And again, not going there. So we'll just say they're all at the same time. If you don't like that, I can show you a bunch of papers, but we're going to say it's at the same time right now. And this thing that happens is that this B cell, when it's proliferating, turns on an enzyme. And this enzyme makes changes to the B cell's DNA. So not only are we going to have this B cell make a clone army, we are also going to make changes to the DNA of this B cell. And so when we make changes to DNA, that's a mutation. These mutations are not happening in eggs and sperm, they're happening in body cells. So that's called somatic. And it's a lot of mutation. It's a higher than average amount of mutation. So it's called hypermutation. So what's happened, so we're getting some changes to the DNA in this B cell while it's proliferating, and these changes to the DNA are called somatic hypermutation. This hypermutation, this huge amount of mutation, is not happening everywhere in the genome of the B cell. It's happening in a super restricted location. So there's only one place in the B cell's DNA where the mutator enzyme is able to act. And that one place is in the VDJ region. Basically, whatever, in this case it's a light chain, so there's a V and a J. If we were in a heavy chain and we had a V, a D, and a J, whatever combo of V, D, and J, and N and P nucleotides we have, that's the only place where the mutator enzyme will act. So you can see basically this is showing you how frequently there's a mutation. There's a bunch of them in this new place where the DNA has been joined, and outside of that there's really no mutations. And so what will happen is that this will be our original gene that encoded the original antibody, and we'll pick up some mutations, and these are point mutations. So just one base pair is changed, or here you can see 3 random base pairs not next to each other, just 3 random point mutations changed. So you're going to have these 3 mutations in the DNA that encodes the antibody gene. If you make some changes in the DNA that encodes the antibody gene, what's going to be the end result? Don't overthink it, like simple answer at first. Yeah, Emily?
Will you not be able to bind to your epitope anymore?
Maybe. Go a step simpler than that. If you change DNA, you change the...
You change the BCR?
Or the protein. Yeah, you change the protein, right? So we're going to change the DNA. As a result, the protein's going to change, right? The B cell receptor and the antibody that's encoded is going to change. And it's specifically going to change right at the part where we bind antigen. So you can see that maybe at the beginning, it looks like this. So it binds antigen kind of mediocrely. And maybe if we pick up some point mutations and we change the protein, now it's going to bind really, really tightly. So that change in the antibody is called affinity maturation. So we're changing the affinity of the protein because we have mutated the DNA. We've done somatic hypermutation on the DNA to result in affinity maturation of the antibody. So this image shows you a situation where we go from kind of mediocre binding to the antigen to a situation where we go to really good binding to the antigen, right? Is that a possible outcome here? Yeah, absolutely. Emily suggested a different possible outcome. What was Emily's outcome that she suggested? Doesn't bind to the epitope. Basically, you destroy the B-cell receptor. You destroy the antibody, right? Maybe you bind to a different epitope. Maybe you bind to no epitope at all, right? So maybe this good situation happens. Maybe a bad situation happens. Could you imagine a neutral, no change situation happening? Yeah, because in some cases, the same base pairs give you the same amino acid. So maybe it actually doesn't change the protein at all, depending on what happens with that point mutation. So you could have lots of different outcomes here, right? Good, bad, zero. Remember that this is happening when the B cell is proliferating. So this isn't happening in one progeny B cell, one clone army child. This is happening in hundreds of clone army children, all at the same time. They're all picking up random point mutations. So the idea is the Emily B cell is picking up some mutations randomly. The Nadia B cell, you all started out with as identical B cells. And now you're all picking up slightly different point mutations through this random mutation process to your antibody combining site. Okay? Or your antibody binding site. Probability-wise, some of you are going to have the scenario Emily mentions, where you destroy your binding. But probability-wise, especially if we go from more than 20 people, like in this class, to 1,000 B cells, somebody is going to hit the lottery. Somebody is going to hit better binding, right? So then what do you want to do after you have this mutation process? Yeah?
Do an additional proliferation of that specific B cell.
Okay, kind of, yes. Yeah, Miles. You want to have a selection process. Now you want to go back and test them all and see which ones bind antigen. And if you think about it, there's a limited amount of antigen, right? So who's going to bind the antigen? The one that has the best affinity, right? The one that won the lottery. The one that did a really good job of mutation. Whatever one got the best affinity for the antigen is going to win in terms of getting antigen contact and getting selected, as Miles points out. And so basically what you do is you make this huge diversity of all your clone army members each get a little bit of variation. And then you're going to test them again and see which one still binds antigen and who's best. What do you think is going to have to happen? What do you think is going to happen here just based on your knowledge of immunology? So we're going to see who binds best to antigen here. Yeah, Paige. Okay, so one idea is you could deactivate the other ones, but there's actually something else that could happen. Yeah.
You could probably death by neglect.
You death by neglect the other ones. It's basically you're competing. Who can bind the antigen best? The ones who don't bind the ones who've ruined their receptor basically are neglected at this point. The ones that have made it a good receptor win the antigen. And so there's sort of this process where we are going to retest all these B cells and see which one binds the antigen. And basically what we're seeing here in this process in the germinal center reaction is we're watching selection or survival of the fittest. We're watching Darwinian evolution at the level of a cell in an organ in your body. So basically you're making a great big population of this clone army of B cells. You're going to make them all vary, and then you're going to just select the best ones. And so while, yeah, some B cells, just like Emily mentioned, are going to do this process wrong, and it's going to suck for them. In our population as a whole, we're going to get better. We're going to evolve towards better binding in the population as a whole. And so this is exactly what happens in the germinal center reaction. Our B cell is going to undergo some mutations in the VDJ region, in the region that binds to antibodies. So here you can see, here's our B cell. It interacts with the T cell. Then it proliferates and does its mutation process. Now that B cell is going to try to get a signal from T cells and FDCs again. This is why it was so important that we were in a place where there was an FDC. Basically, the B cells are going to compete. Who can bind to antigen and who can bind to T cells still? Who can still get pats on the head and not get neglected? If the B cell did a good job in this process, that B cell is going to get signaled again. And the B cell is going to sort of get to go further in this process. If the B cell ruined its binding, it's going to be basically neglected and it's going to die. And so we're going to move the population of B cells towards better binding to the antigen. What's really cool about this process is that the B cells don't do this process once. They will do this process over and over and over again. And so what we tend to see is that our B cell will proliferate and mutate. And it doesn't really, you can't really show mutate here, but it's proliferating and mutating. That's why it says SHM. And then it goes and tests with a T cell and an FDC again. And if we get some signal, then you go back into the dark zone, proliferate again, and try to get even better. Have now a second generation awesome antibody progeny. And then you can do it again. You could have tons of generations where you're just getting stronger and stronger and stronger binding to this antigen. You can see that all of that proliferation and mutations happening in the dark zone, because it's dark, because there's a lot of cells. The testing is happening in the light zone, because we're not seeing proliferation there. We're not seeing nearly as many cells going through this process. Jazz had a question, Miles had a question, and then remind me, I have a point. Yeah.
So is it like I guess a happy medium is like who binds best the fastest?
It seems to be who binds best the fastest, yes.
Okay. So is there like a give and take in a sense where like you can bind like 75% well, but you're binding like within a good amount of time?
I am not sure. I am not sure that we have experimentally figured that out yet. Miles.
Does this involve active killing or just that?
It's neglect. It's not an active killing. So it's just basically it's cytokine withdrawal and signal withdrawal. Yep.
How good does the signal need to be for it to move on before, instead of like cycling back to?
Unknown. We don't know why it stops cycling and leaves. That is another hot area of debate. I'm on the fence about what paper we're going to talk about next week. There are a few different papers that are like right here. This is also like an area of stuff I really find interesting. I will tell you that this particular part of immunology and some of the questions you guys are asking has probably moved further forward in the past 5 years since COVID than it had in the 50 years before that. We have suddenly started to answer all sorts of these. Some of these questions actually may have been published and I haven't read it yet. Like that, we're like doing this stuff all the time. So for example, if you had asked me before COVID, definitely, how long a germinal center lasts, how long you can see cycling with a particular antigen. I might have said, I don't know, like a week, a couple days, I don't know. But like I would have said something short like that. People are now actually experimentally measuring them and seeing that they go out to 6 months. And in some cases even longer. That's actually, so this is part of the big debate in immunologists about if you get a COVID first shot and a booster, whether you should do the same arm or a different arm. Because the idea is if it's the same arm, it's going to go to the same lymph node, and you might actually re-hit a germinal center that was still going and get it to be like a turbo awesome germinal center. Whereas if you go in the other arm, you're going to get a different germinal center. There's actually like papers that argue both sides of same arm versus different arm. But this is all stuff that people are just now getting cool techniques to work out. So some of your questions, like your question, Chrissy, about what the signal is that makes it leave. Also, like I said, there's sort of this, what is the exact order of events in a couple places, are things people are actively working really hard on right now. So this is sort of one big part of the germinal center reaction. As I said, there is another event that's happening along with this proliferation and this somatic hypermutation. Again, does it happen the first time? Does it happen right before it leaves? It's somehow happening in here. It's happening through all this process before it leaves. And the exact details, big matter of debate. But there is this one other process that's happening. And in fact, this is another change that is happening with our B cell. And so what can also happen is that the B cell can actually switch from making IgM and IgD to some other class switch, or to some other isotype. So you can see that basically our B cell interacts with a T cell, an antigen, it proliferates, and then it's going to do things like have maturation of antibody binding, so our affinity maturation. We're also going to have class switching, and we're also going to do things like have that cell become good at making antibodies or good at being a memory cell, but that's a next lecture problem, not a today problem. Today we just care about the class switch and the affinity maturation. And so the idea, if you remember, is that the VDJ region that makes up the variable region of the antibody gene has the 5 different constant regions afterwards, M, D, G, E, A. Remember that M was really close, so it was easy to make an IgM antibody. D was also pretty close, so we could easily make an IgD antibody. And then the other ones were kind of far away, and the polymerase can't make it that far. The polymerase gets too tired. So we never actually make an RNA that has all these other ones. That early B cell never has a chance to make IgG or things like that. What we see happening is that when our B cell is activated, I don't like the order of the next 2 slides. I'm switching them. So the way that this works, the way that we get this class switch is actually that we again see something happen to the DNA. So we're again going to modify the DNA. So we've got another event where the DNA is going to get modified. And what actually happens is if we look at each of our constant region genes, so here you can see the IgM constant region gene, here you can see the IgGs. They don't show it in front of D, but there actually is one there. But don't get stressed about that. Technically, yes, there is one. If we look at this thing in front of M, look at all of these other ones, we see a little sequence that's labeled as S. That's known as the switch site. So we have this little DNA sequence known as the switch site right in front of each constant region. And the way that we have class switch happen is we have a DNA recombination process where we basically have the switch site in front of M and the switch site in front of whatever isotype we're switching to undergo recombination, cut out the intervening DNA. So in this case, we're trying to make IgG1. So we would have recombination between the IgM mu switch site and the G1 switch site. They do recombination. We actually cut out the DNA in between. So we cut out IgM, IgD, in this case IgG3, and we put the IgG constant region right here next to VDJ. So now the enzyme doesn't have to do transcription for a long way. We've basically moved the useful constant region right next to the VDJ. So now we have VDJ IgG1, and we've deleted all that intervening DNA by changing this intervening DNA through recombination, the cutting and pasting of new DNA. And so we see this class switch recombination happening to the DNA where the switch sites are influenced. And so basically you can see kind of the switch site before IgM and the switch site before whatever else sort of interact. We get this new product, and we bring this other constant region gene close by. We delete out the intervening DNA. That, of course, means that we can never go back. Once we have switched this to IgG1, we can never go back to M. We can never go back to D. We deleted that DNA. We could maybe go forward to some of these later ones, but we can never go back because we've actually changed the DNA. And the way that the B cell decides which isotype it's going to switch to is based on what cytokine did the B cell receive. So when the B cell's interacting with the T cell could provide different cytokines. Whatever cytokine the T cell provides, that tells the B cell which isotype that it should switch to. So there are certain cytokines that will induce switch in certain ways. For those of you who've taken molecular genetics, this is probably related to the cytokine changing chromatin accessibility and changing which DNA is able to participate in the reaction based on the cytokine signaling. But if you haven't taken molecular genetics, don't get stressed about that. Yes?
So there's also T cells in the germinal center.
So there are also some T cells in the germinal center. They need to provide T cell help when the B cell's testing. So the T cells are in the light zone, allowing for the help. Great question. Yeah?
These cytokines, is
Mhm.
that the signal 3?
It is the signal 3. Yes. So this is the signal 3 cytokine. It's being made by the T cell in the light zone of the germinal center, exactly as Emily and Paige are asking. So awesome job being on top of that. So this is sort of a big part of what we know about this process. We also know that if I took a lymph node out of you right now, or if I took a spleen out of you right now, and looked at it under the microscope, we would find germinal centers. You can actually find germinal centers under a microscope really easily. Oh, maybe I'll do that paper. I forgot about that paper. That might be a good paper. All right, I got to decide about which of these papers are. Now I got like 2 in my mind, and they're both pretty good. If I looked at you right now, I could find germinal centers, because you're always making some response to something. Ideally, you're going to, then kill the something and not ever feel sick. But we can always see germinal centers in you. It also turns out that we can find some patients who don't have germinal centers. And so there are some patients who do not have germinal centers. One thing that happens if you don't have germinal centers is you don't get affinity maturation. So all your antibodies are kind of mediocre binders. You don't get the really strong binders. That's sad. The other thing that happens is that you can never make IgG, IgA, or IgE. You can never class switch. You are stuck making IgM for the rest of your life against every antigen. This disease should actually come over here so I can change the slide. This disease is called hyper-IgM syndrome. So basically the idea is that people are making 100% of their antibodies are IgM. That kind of looks like you have too much IgM, but it's really because you have 0 of all the other ones. And you can see this is actually a normal lymph node. You can easily find germinal centers in normal lymph nodes. This is somebody with hyper-IgM, where we see no germinal center formation, and thus this person is always going to have IgM and IgD. They're never going to have GA or E. They're also going to have very mediocre binding antibodies. These people have are pretty severely immunodeficient and are pretty sick. This is not a great disease to have. Yep.
Wait, so if the germinal center is defined by where the B cell goes, then how is it?
So part of it, why does the B cell go somewhere to make a germinal center? Why, Paige? Because of the T cell, right? So that can start to lead to a little bit of, I can answer your question partially, David. So part of your issue comes from thinking about the fact that we actually know there are 3 different genes that could be mutated in patients with hyper-IgM. So 2 of the genes will sort of address your issue. The 3rd one, you just got to believe me. Okay? It turns out that the 2 first described genes that are involved in hyper-IgM syndrome are either patients who don't make CD40 or patients who don't make CD40 ligand. So basically you're getting this defect because the B cell can't interact with the T cell, just like Paige said. So either the partner on the B cell is missing or the partner on the T cell is missing. So you can't get the interaction between them. And so in that case, David, the B cell isn't going to move to a germinal center because it can't interact with the T cell. It can't get signal 2 from a T cell to move to the germinal center. And so that is sort of part of how this happens and part of what goes on with hyper-IgM. Like I said, there's a 3rd gene. I can imagine a complicated way that things could work with a 3rd gene that is way beyond where we're talking about now, but if you really want to sort of debate it through, we can do that at a later time. But basically this sort of is the process we've talked about at this point. So we have our B cell that comes out of the bone marrow. It has IgM and IgD on its surface, gets to the lymph node and sees antigen as well as a T cell. That B cell is going to undergo clonal expansion, make a whole bunch of copies of itself, and is then going to do class switching as well as affinity maturation, this sort of mutation process. Yes, there are these other 2 pieces of it, but those are, like I said, those are Friday's problem. They're not today's problem. When I started graduate school, this was the end of the story. But this is not the end of the story anymore. Because it turns out that right at the beginning of my time in graduate school, you guys read a paper actually about one really important immune gene that was described like basically in my first year of grad school, which was FOXP3. So like I'm a first year grad student who doesn't know anything, and then suddenly there's FOXP3 and there's Tregs out of nowhere. Also that same year, AIR, remember from T cell development, was also discovered. And another gene was discovered. And this gene is really important in the germinal center reaction and understanding this process. And so now we understand a lot more about how exactly some of these events are happening in the germinal center. We've got these 2 events that we're seeing in the germinal center. One of them is class switching. One of them is affinity maturation, which is happening because of somatic hypermutation. There's something besides they happen in the germinal center that these 2 things have in common. So there are 2 things there's this other thing that these 2 processes have in common. What do these 2 processes have in common? Yeah, Emily.
They're like cutting up DNA
they both involve something going on with DNA, right? There's something happening to DNA in both cases. It's either the DNA at the switch site or the DNA at the antibody combining variable region, right? What was discovered here is actually it's the same enzyme that messes with the DNA in both cases. So there's one enzyme messing with DNA at this point. The way that it messes with the DNA varies a little bit in terms of whether it's going to lead to class switching or whether it's going to be affinity maturation. But we got one enzyme that's going to do basically the results of give us both of these things. And so this enzyme was also discovered during my first year of graduate school. So before that, this all happened by magic, but then we found the one enzyme. And this is also one reason why this gets sort of, tricky to study, because it's hard to figure out exactly which event is happening first when it's one enzyme doing them both. This enzyme is called AID. When I tell you what AID stands for, you're going to be annoyed. Because AID stands for something that has more than 3 letters. So AID is activation-induced cytidine deaminase. We don't put the C in the abbreviation. And the reason why we don't put the C in the abbreviation is because AICD means something else in immunology that we haven't learned about yet. So AICD was already taken when this enzyme was discovered, so they went with AID instead. But one of the things that you can see in the name is that we've got cytidine. You can also see I have it as cytosine at the top. I know Dr. Casano would care a lot about the difference between the two of them, and I am not going to get super into that right now. But you can see an example of cytidine here. What is, he would probably care too. What is cytidine? Where have you heard of cytidine before? Or cytosine, either of them? What? It's one of the 4 base pairs of DNA. This is C. This is your C of DNA, okay? You can see that this cytidine has this amino group here, right? So it has this NH2 as part of the chemical structure. If you were going to remove an amino group from something, what would you call that reaction? Yes?
Deamination.
You would call it deamination, de-aminoing, deamination, right? So what AID does is it deaminates cytidines. So basically the idea is that this is an enzyme that's present when the B cell is getting activated, and this enzyme is able to deaminate cytidines. So it's able to take this amino group off of cytidine. Now, you might say, okay, great. Who cares about when a cytidine gets deaminated? But this image shows you why you care about a cytidine becoming deaminated. So here you can see the sort of before the cytidine. Here you can see the after. What is this product that we're seeing as the after of cytidine deamination? So when you deaminate a cytosine, what do you get? Yeah?
A deamination of the amine
Mm-hmm.
leads to an oxygen, which is uracil.
Yeah, you make uracil. So the product when AID acts on a cytidine is that cytidine becomes uracil. What's uracil? Yeah.
It's the base pair in RNA.
It's a base pair that's in RNA. It's U. It's only in RNA. And basically what we're having happen here is we're taking the C, a C in a piece of double-stranded DNA, and we're changing it to a U. That is a problem. And it is a problem for 2 reasons. One reason Julie kind of already told us. This is a base that's supposed to be in RNA, and we just put it in DNA. We just did a chemical reaction that made this base be in DNA, and it's supposed to be only an RNA thing. That's not good. What's the other problem here? Yeah.
That is supposed to bind to guanine,
Mhm.
but now it's the uracil, which is supposed to bind to adenine.
Yeah, so here's the thing. This is happening in DNA that is already paired. Like this C that's getting reacted is bound to a G somewhere. And we just turned that C into a U. G doesn't go with U. That's wrong. So we got the wrong pairing, and we got a base pair that's supposed to be an RNA. This is not okay. And so what do you think is going to have to happen because this is not okay? What do you think?
It has to get like taken care of. It has to get chopped.
It has to get taken care of. We got to do something, okay? We got to deal with this. Officially, we have to resolve the uracil. We just took this and we changed that C to a U, and now we got a UG base pair in the DNA. That's bad, right? So there's 3 different things that could happen. Well, one of them is DNA replication could just happen there. So maybe we don't deal with it at all. Maybe like just as luck would have it, we replicate before we get a chance to deal with it. When that occurs, this piece of DNA that has the GA gets a new CT partner. Is there any change to that DNA from the parental? No, you basically get back what you started with. Womp womp, no one cares. But we also, if you do that replication, are now going to take what was originally CT and GA like this and turn it into TTAA. So basically, when the polymerase comes along, it's going to see the U and it's going to be like, I know what to do here. I put an A here. And so it's going to change this from a CG base pair to a TA base pair. That's a point mutation. And so basically, if we replicate this DNA before we fix the uracil, we just got us a point mutation, like we might need in somatic hypermutation. We just somatically hypermutated. We can also try to deal with this U. One way of doing that might be to cut out just the base part. So you can see we cut out just the U. And when that happens, we actually generally fill in with something else. So we kind of fill in with whatever other base there is around. It could be it could be a T, it could be a G, it could be a C. It could be anything. When that happens, there's a 1 in 4 chance we get back what we started with, right? But there's a 3 in 4 chance we get something new. We get something different than what we started with. That's a point mutation, just like we need in somatic hypermutation. So in fact, this uracil, 2 of the different ways we can deal with it, both give us a point mutation in the resulting DNA, and both basically are giving us a somatic hypermutation. One other thing can happen if there are a bunch of U's in a row next to each other. If there were a bunch of C's that all got turned into U's in a row, we have to cut out a big piece of DNA. This is only showing us cutting out 2, but the idea is that we're cutting out a big piece. Yes, if you took molecular genetics, we could go into much more detail about this, but I'm not doing that right now. We're cutting out a big piece of DNA. That DNA is pretty well damaged. And so the only way that we can repair that very damaged DNA is to find a similar piece of DNA and do recombination. Basically, we're going to fix it by stealing some similar DNA from elsewhere in the genome. If we have those point mutations, we're going to end up with somatic hypermutation. So either of the first so if we have that cytidine in the variable region of the antibody gene, we're going to get hypermutation. If that cytidine that is reacting is in a switch site. Usually there's a ton of Cs in the switch site. The switch site gets super damaged. And the only way to fix it is to merge it with another similar piece of DNA, aka another switch site. And so basically what we'll see is our IgM switch site will get super damaged, and we'll see recombination with another intact switch site, whichever one is available. And that will be what gives us the recombination for class switch recombination. So both of these processes are happening because of the DNA damage happening as a result of AID's action, deaminating cytosines. And so you can see this overview of this process here. I will say a bit more about B cells next time as we go into kinetics and memory. Just so you know, there's a slide that looks, it's slide 38, it looks blank. But actually, if you go to it, it's a video of this whole process that we just talked about. So if you'd like to watch exactly what we're seeing right now in a video form, go to slide 38.